All right, so for those of you that don't know me, my name's Doug. I joined the BRC earlier this year. Today I'm here to give Matt and Tom a little bit of a break and to talk about ChIPseq. So let's see, let's pull up the, so this is our normal training page, the same page you guys have been accessing throughout the entire course. As usual, we'll click on the course, the relevant course. So, you know, hopefully you guys have obviously installed a lot of this stuff, IGV. This is our package developed in the BRC to get external packages that aren't in R, and we'll use that today for our peak caller max 2, and then all of the relevant packages for today's lecture, as well as the material. So hopefully you guys have downloaded that so you can follow along with the code if you'd like. So just overall today, we will first spend some time this morning looking at pre-processing and alignment before moving on to some quality control and peak calling, and then finishing the day in the afternoon with some functional enrichment, motif enrichment, as well as identifying high confidence peaks in differential ChIPseq. And so we'll get started with lecture 1. Okay, so just the briefest of all introductions for ChIPseq for anyone that has not done it before. So ChIPseq is short for chromatin immunoprecipitation followed by deep sequencing. So the idea is to look for enrichment of a certain protein or transcription factor binding, or some sort of post-translational modification on the histomark, for example. So the basic idea is that you're going to take your chromatin, fractionate it in some way, whether it's through sonication or some sort of enzymatic fractionation. Sometimes you cross-link your proteins to DNA, and then you enrich certain DNA binding sites or epigenetic marks with an antibody binding to your protein of interest. And then once you have your fragments of DNA that you're interested in analyzing, you go through some process of library prep. Usually this is preparing for Illumina sequencing, where you eventually want to get adapters on your fragments that will both allow you to PCR your fragments and sequence them on the Illumina sequencer. And these fragments can be either sequenced from one end or both ends. For ChIPseq, it's usually not necessary to do paired end sequencing. Usually single end is enough, and it's generally cheaper. So, you know, if you don't need to do paired end, you typically won't. So for ChIPseq, we're usually getting single end reads. So like most of your other workflows that we've gone through in this course, the raw sequencing data coming off the sequencer is going to be in Fastq format. And you've gone over this before, but just to reiterate, the Fastq is going to have some, it's generally repeating 4 lines. The first line is going to have some identifying information about that read. Then your actual sequence, the third line is typically blank, and then a fourth line that contains some quality scores about each particular base in that sequence. Just to go over a little bit the data that we're going to be using today in this workshop. It's all retrieved from the ENCODE website. It's basically, there's 2 replicates of ChIPseq for the transcription factor MYC from both the MEL cell line and CH12 cell line. I believe these are MEL is a leukemia and CH12 is a lymphoma cell line. We can get the raw sequencing data from ENCODE, as I said. This link will bring you to the information about replicate 1 of the MEL cell line. Here, you can click on this and it should directly download it. And the same links for the second replicate are here. So working with raw ChIPseq data, the Fastq file, this is going to be relatively similar to, in some ways, to what you did with Matt yesterday in RNA-seq, but we'll quickly go through it in terms of ChIPseq. So we're going to use the short read package, which allows you to both bring Fastq files into R and also interact with it pretty easily. So we'll load the short read package. And the first thing we'll do is look at some general QC. Again, this is pretty similar to what you did yesterday. But we will also, because R isn't great at handling large files, we're only going to subsample a portion of the Fastq file. And this is generally actually fine. Most QC packages that you'll interact with really only do a subsample, and that's generally, that will give you a good representation of the quality of the entire sample. But it is important to note that when you subsample, and most of the packages that subsample Fastq files will do this automatically, but it's important to retrieve random reads across the entire Fastq. So the Fastq file is ordered by the position on the sequencer, and there could potentially be some kind of bias depending on where the sequence is sequenced on the sequencer. So we can subsample the Fastq file with the short read package using the Fastq Sampler and yield function. So this will randomly sample a defined number of reads from the Fastq file. So this is our function here. It just simply takes the path to our Fastq and the number of reads you want to sample. And this just kind of sets it up. And then once you use the yield function on this object, it will know how many sample or how many reads to take. And it will take that randomly from the Fastq file. And so once we've done this yield function, we get this this R object called of a class short read queue. And so this object is something that we can use short read functions to interact with, and allows us to pretty easily assess quality. But it tells you the number of reads and also the number of cycles for each read. So the length of each read coming off the sequencer. And so I think you guys went over this yesterday a little bit, but these are some of the common and familiar accessor functions that we can use within short read to look at our Fastq reads that we just read in with the yield function. So we can look at the actual sequences. We can get an object that has the ASCII quality scores, and we can also get the IDs. This is essentially just accessing different lines of that 4-line Fastq file that we saw earlier. And if you just print out the result of the sread function, it's this DNA string set object, which is a nice R-looking object with each line. It tells you the width and the actual sequence of all the reads that you just sampled or that you read in. So we'll now check some sort of basic quality scores of our subsampled Fastq data. So we can use the alphabet score function with the output of this quality function, and this will give us the sum quality of all of the bases in our read. And then we can easily plot that with ggplot, where we just turn it into a data frame that just has all of our read qualities, or all of the summed read qualities, actually. If we make a histogram, we can see we have, most of our reads are piled up over here at around, I don't know, 11 or 1200. And so if you think about this, this corresponds to quality scores around 30, which is good. The quality score is basically the probability, or it indicates the probability that base was called correctly coming off the sequencer. And just for reference, a quality score of 30 is basically it will be wrong 1 out of every 1,000 times. And so that's we're pretty confident that most of the reads out here are high quality. But you do see we do have this population of really low quality reads. And this is something that it's good to be aware of. It's common, and we're gonna soon try and filter those out, because you really don't want a lot of low quality reads in your downstream processing, because it can throw off things like alignment and peak calling. We can also look at the frequency of different nucleotides, both within each read and across cycles. So here we use the alphabet frequency function to assess the number, or the proportion of each read that has A's, C's, G's, and T's, and N's, which N's are bases that were unable to be called. So we take the results of the sRead function, which is that DNA string set object, and we apply the alphabet frequency function. This gives us a matrix that has the nucleotides as columns and the reads as rows. And we can just simply take this matrix, do call sums to get the sum of all of the A's and C's, G's, and T's, and we'll do N's as well. And you can see you have a higher number of A's and T's than G's and C's, but at least they're each similar. This is common in the human gene, or in the mouse and human genome. You typically have less G's and C's in your sequencing data. We can then do the alphabet by cycle function, which will give you a matrix that is the rows will be the nucleotides, and the columns will be each cycle. Here we have, since it's 36 base pairs long, we have 36 cycles. And we will turn this into a data frame that we can visualize. And if we plot this data frame with ggplot, we get a nice visualization. You've probably seen this before in some of your QC outputs from your own sequencing. And again, we have slightly higher levels of A's and T's than G's and C's, which is common, but you basically just, you want this to be constant across the cycle. You don't really want much bias on either side. And a lot of these things, I think it's important to note that in isolation, each sample, it can be kind of hard to assess whether, this is good or bad or whatever, and oftentimes when you compare them across the many samples or the multiple samples in your data set, that's when you can really start to identify whether something seems off. And I think that's a little more useful than, you have to build these individually, but then when you look at them side by side with their samples, you can identify outliers and maybe correlate things that seem off in measures like this with some downstream, like a lack of peak calling or something like that. We can also look at the quality across cycles. So we can easily turn this quality object, which is just, if you look at that, it's just going to be the ASCII scores for each base. We can use the as matrix function to translate these ASCII quality scores into numeric scores. So you have a matrix that's your, the rows will be your sequences or your reads, and the columns will be the quality scores at each position along the cycle. And this is, if you just plot this with base R, you can see a pretty common pattern where as your cycles progress towards the end, your quality decreases a little bit. You can see this both in the mean quality and also the sort of distribution. You're getting a larger distribution, but this is common. It's definitely common that towards the end, your quality goes down a little bit, but you can see this doesn't dip very much. You don't even really get below a mean score of 30, which generally above 30 is really good. 20 is pretty good too. So this is common, but if this dropped off a lot, and again, if you're seeing one sample that's much worse than the others, that's a reason to at least be careful with that sample in downstream processing. But this looks pretty good. So overall, these quality scores and distributions look pretty good. And as I said before, in isolation, it can be hard to really evaluate a sample, but when you look at them together, it makes it much easier. But we did observe the second population of low quality scores. So we are now going to work to remove those, both those with low quality scores and with high numbers of unknown bases. So as I think we've talked about throughout this course, handling large files in R can sometimes be difficult, and Bioconductor has a lot of functions that sort of cater to this and allow us to interact with these large files. One of which is Fastq Streamers. So this is different from the function we had just used, which was Fastq Sampler. Fastq Sampler will take a random sampling of your reads. Fastq Streamer actually is cognizant of the reads it took before, so it's able to go through your entire Fastq file and get through the end without, retaking certain reads. So if it takes reads 1 through 10 the first time, it's going to know that and go to 10 to 20 the next time. And it just simply takes a path to your Fastq file and the number of reads you want to take with each yield. So here we're just going to set up a loop, a while loop, where we will basically go until the yield function yields no reads, meaning that we've reached the end of the Fastq file. We're going to, in this loop, we'll catalog the total number of reads we go through. We'll first filter out the reads that have, or we'll filter out the reads that have a quality, some quality above 300. So we'll get rid of the ones that obviously are below 300. Then we'll take those filtered reads and then filter out the ones that have more than 10 uncalled bases or more than 10 ends using the sread function, or the alphabet frequency function combined with the sread accessor, which just gets the sequence. So after applying those two filtration steps, we have our filtered reads, and these are just the filtered reads within the particular yield that we're going through. But we can use the write Fastq function to write to this path in this mode A. It means append, so you're just appending to this file that's already there, so it won't overwrite it. So as we go through this while loop, we'll just continually keep adding the filtered reads piece by piece until we have our complete Fastq file that's filtered. And so once we have our filtered Fastq files and we know that the QC looks good, we can move on to alignment. And this will be similar to what you guys did yesterday, although ChIPseq is continuously aligning to the reference genome. We don't obviously have to worry about like exon junctions like you would for RNA-seq and stitching them back together and being splice aware. So it's a little bit simpler in terms of the algorithms of the aligners, but any of the aligners that we've seen in previous sessions, and you can link to them here, will work here. We'll go through a couple here today. But the output of any of these aligners is going to be a BAM file or a SAM file, but the BAM is, as you learned, is the binary version of the SAM file, and the SAM file is the one that is shown below that is kind of more human readable. You guys learned about this previously, but just quickly, there's some identifying information in the first line and also some important flags in the fourth line that you can go through and look up both programmatically and just anywhere if you Google it. The second line is going to be the align sequence, and the third line will be quality scores or scores that indicate how sure we are that this base actually aligns to the location that's indicated in the cigar string. And so before we align, we need a reference genome to actually align to, and R and Bioconductor make that easy to access. These have all been curated and put together for us in nice Bioconductor formats. And we'll do this through these BS genome libraries, which we you've hopefully downloaded, and you can just load into your R session with the library function. If you look at it, it tells you where it came from, when it was released, the species, which build of that species it is, and then the different sequences or chromosomes that are available. And so what it is basically just a list that, and each element is a different chromosome. So we're gonna not worry about the random and unplaced contigs. We're just gonna look at the major chromosomes. So we'll make a vector of the chromosomes that we're interested in, and we'll use lapply to just cycle through this BS genome object and only access the chromosomes that we're interested in, which is for the mouse 1 to 19, XY, and the mitochondrial. We'll give them the names, sensible names of the chromosomes, and the output is this DNA string set object after we've used this DNA string set function on this list that we extracted from the BS genome object. And we have a DNA string object, a string set object that has all of the chromosomes that we're interested in. So each of these list elements will just essentially be one long string of sequence for each chromosome. So now we have this DNA string set object. We can use the write string set function to then write this to a FASTA file on our local computer. And this is what we can use for our alignment. And so we're going to be aligning using the subjunct algorithm, which is coming from the R subread package. And as is the case with pretty much all aligners, before you start alignment, you'll need to first build an index, which basically just provides, like the index of a book, it kind of provides a map for the aligner to more quickly access the FASTA file as it tries to align your reads. So for our subread, it's just the build index function will allow us to do this. And so you guys should have done this for RNA-seqs. You shouldn't have to do it again if you're going through the code, but building an index is pretty memory intensive. And you can control the amount of memory that is going to be used during the process with this memory argument. And that will change depending on your laptop, sort of how far you can go. But it's just kind of something you have to play with. But this can take a while and can make your computer pretty angry if you're not careful. But this function basically just takes the name of the index that or the name that you want it to be. In the end, once it's going to create a number of files, and it'll append this name to it. It'll take your FASTA file, your memory, and also whether you want to split it up as it's being made, which can help with the processing as well. And so once we have our index and we have our FASTA file, then we can actually align our reads from the FASTQ to the reference genome and create our BAM file. Let's see, so with the align function from our subread, it takes the name of our index. It'll take our FASTQ file, the output format, which will almost always be BAM, the name we want for our output file, the type, and the offset, which this one's 64. I think more commonly recently it's 33. It's just the way that the quality scores. There's two different modes of ASCII scores. It's either going to be 33 or 64. And upset, our subread will get upset if it's wrong. And there are ways to check whether that's correct or which one you have, but just make sure you know and put that in correctly. And then the number of threads is how many cores you want to use in parallel. So this can all be done in parallel. And so this will depend on how many sort of open cores you have on your computer. If you're worried about it, you can just set this to 1 and it will do them sequentially. But if you think you can use multiple cores on your computer, this is probably obviously more relevant for interacting with the HPC, where you can use lots of threads, and this will decrease the alignment time pretty significantly. And so another common aligner that you may have heard of is Bowtie, and you can interact with Bowtie within R as well. There's the R Bowtie 2 package for the most recent Bowtie version, Bowtie 2, and the Quasar package allows access to the original Bowtie aligner, but that is a little slow and memory intensive. But we do have a session dedicated to this, so you can look at that if you're interested. But we'll go through how to use the Bowtie 2 aligner here. So we'll load it into our so we'll load our Bowtie 2 into our session. And again, we have to build an index, and this is done with the Bowtie 2 build function. So this just takes the FASTA file, and then this is going to be the output name that we want, again, appended to the index files. And that will take a while. So once that's done, Bowtie 2 is a little bit different from our subread in that it needs uncompressed FASTQ files, and it also outputs a SAM file as opposed to a BAM file, which is fine. It just results in some more kind of larger intermediate files, which you can easily just delete them. But this is kind of something to be aware of, especially if you're using this in a pipeline format where these files are generated and can really balloon the amount of space you're taking up. And so it's just something to be aware of. And if you're doing it a pipeline, you probably want to be conscious of that and delete some of the intermediate files as you go through. But so first we will, using the gunzip function, unzip our FASTQ file. And then that's what we'll use here, the unzip version in our Bowtie 2 alignment call. And so this just takes the index, the output of the SAM file that we want it to be called, and our FASTQ file. And again, the output is a SAM file, so we need to convert it to a BAM file, which we can do with the RSAM tools as BAM function. And as I said, an important consideration with our Bowtie 2 is these sort of larger uncompressed files. And on the command line, which is where a lot of people are using our Bowtie 2, you can actually stream them. So you can stream the output of your alignment to directly make a BAM file, never actually have the intermediate. You can't quite do that yet in R. And so again, our Bowtie 2 can create lots of intermediate files in R. But again, you can just easily delete them with this unlink function. So if you are in a pipeline format, you can, once you know you have your BAM file, you can then go back and easily delete the SAM file and reduce the amount of big files you have hanging around. And so once we have our BAM file, we need to sort and index them. And this is largely for downstream processing. So this is going to organize the BAM file in a way and provide the index that allows many external programs to easily interact with it. And so most external programs won't work if you don't have a corresponding index, if your file isn't sorted and if your index isn't sitting in the same directory. And this includes IGV. And so we'll use our SAMTools functions, sort BAM, which I think Matt mentioned yesterday, but one little caveat here is that it automatically appends .bam to the output. So all this takes is your BAM file and the output name. But if you did put this as, sorted mel.bam, it'll do .bam, which is a little annoying, but that's how they've organized it. So just be aware of that. And then when you just use the index function, you take your sorted BAM, and it'll just automatically create the index in the same directory as your BAM file. And so looking at and assessing our map data, we can use the, once we have the index and sorted BAM file, we can use the IDX stats BAM function from our SAM tools, which will create a data frame that indicates the number of mapped and unmapped reads. And so if we just want to look at the number of mapped reads per chromosome, we just sum up the number of mapped reads from this data frame, which will have each chromosome, and then we plot that. And we can see, you know, the larger chromosomes have more reads, which makes sense. And so this is just another kind of sanity check that allows you to assess that everything is still looking pretty good. You know, if you had one chromosome where there were no reads, then that would obviously be a concern. And then you could go back and look at your code and try and understand where things went wrong. So once we have our sorted indexed BAM file, we feel good about its mapping. We can use the coverage function to create an RLE list object, which is an R object, and then we can easily convert this RLE list to, or export it as a bigwig with our track layers export .bw function. And the bigwig is the file, or one of the files that you can easily visualize on IGV. So this is typically why we're doing this. It's also important to, if you want to compare, if you want to look at all of your samples in the same IGV session, or even compare, you know, look at counts in some downstream software, it's nice to have a normalized bigwig. So, you know, if one sample was sequenced deeper than another and had more reads, a bigwig is simply going to tell you how many reads are at each genomic locus. And so if you had more reads in your library, you're probably going to have more reads stacked up in the peak regions. And so we can easily normalize these. And one way to simply do it is just take the number of mapped reads, multiply by 1,000,000 to get your reads per million, and then use this weight parameter within the coverage function, and that will essentially divide your coverage by this normalization factor. And this should bring your bigwigs down to the same level, so when you look at them in IGV, they'll actually be somewhat comparable. And so, you know, now we've gone from Fastq to alignment, assess the quality, and now we have a nice bigwig file that we can actually open up in IGV and scan and look around and see if we actually have nice enrichment. So this looks really good, where, oh, we have a really strong enrichment here. And, you know, the concern would be is if you looked at this and you just went across the entire genome and it was completely flat, which, you know, happens for, you know, not very efficient antibodies. And so I think you can get a pretty good idea of how well the chip worked just simply by looking at it. But we'll move on to do some peak calling, which will allow us to actually statistically assess whether this is an enrichment compared to the background in the next section, I believe. Yeah, so that's it for this first lecture. There are exercises here, and as sort of we've been before, as we've been going through this quickly, we're not going to do the exercises now, but it is really helpful for you to go off and do that on your own. And we're always here to answer questions. And I think Matt's mentioned before, there is in the course page, there is a GitHub issues page where you can ask questions if it's not during one of these sessions, and we'll monitor that and answer. So that's the end of the first section. So we're going to go straight on to the next one now, which is some more QC, and then we'll take a break and come back later. So hopefully everyone's still there, and doing well and excited. So we're going to keep going on to more QC and peak calling, and also annotation of peaks. And okay, I think, and Matt, this is how Matt and Tom, I think, have been running it before, but obviously if you have questions, feel free to put them in the chat or just speak up and I can pause and answer them. Okay, so this is the same data that we started to look at in the last lecture. You can get it from ENCODE, or you can download them directly through some of these links. But we're using, again, both 2 replicates from the MEL cell line and 2 replicates from the CH12 mouse cell lines, and these are MYC transcription factor chips. And so now that we have a BAM file, so we looked at QC of our Fastq file in the first lecture. Now that we have an aligned BAM file, we can assess the quality of our alignments. And ChIPseq has lots of potential sources of noise. And it varies not only between chips, meaning like between different antibodies, but even within the same chip, you know, you can have different efficiencies, or the same antibody, different samples, you can have different efficiencies. So it's important to do our best to assess QC and understand whether any of our downstream analyses and results that we see are not due to sources of potential bias and noise, and hopefully actually due to some sort of biological effect that we've studied. Some potential sources of noise are varying efficiency of antibodies. So I don't know if any of you have ever run a Western blot, but in the same way that a Western blot has tons of background, those background bands that you see can also be proteins that bind to DNA. And so you can imagine that there'd be lots of DNA that is not necessarily the DNA that you're interested in, but the hope is that the efficiency of binding your protein of interest is such that you can overcome that background. Similarly, non-specific binding. Also, library prep is another point at which bias can certainly be introduced, and you'll often see that in your aligned reads. And specific to CHIP, there are some artifacts, and we'll go through those pretty extensively today. And because people have noticed this, there's certainly people have developed ways to understand how substantial your artifacts are and ways to get around that for peak calling. Here's some links that if you're particularly interested in some of these QC metrics and QC methods, you can click and read more about. One thing to briefly mention is, especially when it comes to peak calling, is running or having an appropriate input. It's always something you should consider before you start your experiment. But input samples are typically made from fragmented DNA prior to your IP enrichment. And so it's essentially just before you add your antibody, you take a bit of that DNA. And what it allows you to assess is whether certain regions of the genome are going to be overrepresented in your sample before you even do an IP. So that means, but when you do the IP, there's a greater chance that those will come down. And, you know, ideally and usually your input looks pretty flat where there's really no peaks. Sometimes you might get slight enrichment in regions that are more open chromatin, and that's because especially if you're using sonication, more open regions are more likely to be sonicated more efficiently and pop out. And so you'll often see sort of slight enrichment at promoters, for example. Hopefully not so much that they're peaks, but in your IP should hopefully should be much higher than that. But it is important to do that so that when you do, as you'll see with our peak calling, you can actually compare your chip to your input and make sure that your enrichment that you're seeing is actually unique to your IP and not present in the input. You're certainly you should come to us and talk about this if you like before your experiment, but people kind of go about this in different ways. I don't think it's really necessary, but you could do an input sample for every single chip sample that you do. Oftentimes people will do a different input sample for either a different tissue or cell line, for example, and then you can use that same input to compare across different conditions within that same tissue. So if you have multiple treatments, you can just typically use one input for all those treatments. I mean, you can, there could theoretically be a situation where your treatment is so profoundly changing the openness of the genome that maybe you'd want another input, but typically that's not the case. Usually it's just across different tissues or different cell lines you'd want different inputs. And so we will use the ChIPQC package, which accesses and wraps up some metrics for alignment. This is a package that Tom wrote before he came to Rockefeller, so we know it well. And sort of the workhorse of this package is this ChIPQC sample function, which takes your unfiltered BAM file, or filtered, you can do either, but the genome, and then your hopefully a bed file with your blacklisted regions. And so I'm not sure if you guys have talked about blacklist yet, but it's just to go over it quickly again. And this blacklist can be either a bed or a G ranges, but the blacklist is a common artifact within ChIP experiments where for whatever reason, probably multiple reasons, certain regions of the genome just get piles and piles of peaks, and they're not real biological peaks. They don't look like peaks. They're just like these kind of really sharp, giant peaks that seem to exist across multiple tissues, multiple cell lines, any antibody. So it's really just truly an artifact of ChIP. And Ansho Kundaji, who is a professor at Stanford and heavily associated with the ENCODE project, his lab has curated or taken multiple experiments and curated where these common blacklist regions are, and you can go to their site or go to ENCODE and just simply download the blacklist bed file for whatever genome that you're using. And here we're going to use it to assess the number of reads that actually are in the blacklist, because it can be obviously a concern if all of your reads are going to blacklisted regions. So you can directly download that, the MM10 one with this link here. So we can use, again, this ChIPQC sample function from the ChIPQC package to easily evaluate some of these important BAM file QC metrics. And so what we're going to do here is we're going to load the package. We'll have the path to our blacklist, and then we'll run this ChIPQC sample function on one of our BAM files. Here's the name of the BAM, the genome we're using, the path to the blacklist, and if you just want to look at certain chromosomes, which can help, obviously speed things up or use less memory, you can specify the chromosomes you're interested here. And so after you run this, if you look at the class, this is a ChIPQC sample object, which, as you've sort of encountered many times so far, many packages create their own sort of types of R objects, and these are just ways to package lots of information in ways that you can easily manipulate and access through the specialized functions of that package. And so if you just print out this ChIPQC sample object to the screen, you get some sort of helpful information, the number of map reads, the number of reads passing a MapQ filter. And so MapQ is a measure of quality for the entire read for the mapping. And I believe, and you can look up sort of what the different mapping qualities mean, but you can set this filter within that ChIPQC function. I think the default is 15, which is a pretty standard filter. And so you can see sort of how many of your reads pass a pretty standard quality filter. This BAM file actually has duplicates filtered out, and so that's why there's 100% as non-duplicates. And we'll get into some more of this information later on. But anyway, you can easily, right out of the box, just sort of see some interesting metrics for the sample that you made this object for. So we have multiple samples. So we want to, ChIPQC has nice ways to kind of interact with multiple samples and look at them all together. And so we're going to do that here using lapply. So these are the names of all of our BAM files. So what we have is in the end, we have 2 replicates for each cell line, for each MIC chip in each cell line, and then we have an input sample for each cell line. So we have 6 total BAMs. We're going to use BP lapply, which is a bioconductor specific version of lapply that will, if you want, you can let this go in parallel, which will speed it up. That's more relevant on the HPC. But this takes, for the lapply, it works just like lapply. You have the path to your BAM, so it's going to loop over these BAM files, apply the ChIPQC function, and then, like any lapply, these are the arguments that will then be used for this function on this list of files. And so this will give us a list of these ChIPQC sample objects. We'll name this list with the names of our BAM files. And all ChIPQC functions can also work with a named list of ChIPQC sample objects to nicely aggregate these samples into a table, as well as this works for most plotting too. So here we use the Qcmetrics function, which will give us an overview of all of our BAM files and the important metrics for each one. And we'll go into more detail about all of these, but obviously seeing them side by side can really give you a good idea of whether anything stands out. So just this is your mapping percentage, the percentage that was filtered based on the quality score or the quality threshold that you set in the function, our read length, our predicted fragment length. We'll talk about these, but these are some metrics that can help you identify low quality samples. You can also put in a bed file of peaks, and this is the percentage of reads and peaks. And so the percentage of reads and peaks is a nice metric to tell you how efficient your chip is. It's surprising how low this can be, and your chip can still work. Really, really good chips, like something like CTCF, you might have 60% of your reads and your peaks, whereas a less efficient transcription factor might be as low as 2 to 5%, but sometimes that can be good enough to at least get some peak calls. And so, this number will be highly variable dependent on your chip. But for example, if you have a bunch of chips that are all the same antibody, it is a nice way to compare sort of within antibody treatments. And this is the percentage of reads that go to blacklist. So you can see already that this is not abnormally high, that a large percentage of our reads are going to these blacklisted regions. But with that said, there's still many, many reads, 85, 90% of reads that are still available for us to really analyze in a meaningful way. But this is at least a good metric to catch, to understand, if this number was 60 to 80%, that would obviously be a problem. So it's good to at least know. As we mentioned at the beginning, ChIPseq, you're often doing single end sequencing, and so you don't know the fragment length, right? Just from the sequencing, because these reads are 36 nucleotides long, or base pairs long, and your fragments are typically going to be around 150 to 400 base pairs. And so all you're doing is sequencing either end of that fragment, but we have pretty good ways of predicting fragment length based on single end sequencing. And this prediction of fragment length is essential because it's important for peak calling and identifying where the actual middle of a peak is, and the coverage profiles that we use for IGV. And so cross correlation or cross coverage, which is a metric that ChIPQC catches, is important for understanding or for assessing fragment length. And so as we've gone over, part of this process of ChIPseq is to fragment your DNA with either sonication or some kind of enzyme like micrococcal nuclease. And these fragments are the things that you're actually sequencing. And so we want to try and predict how long these fragments were. So ChIPseq is typically, like I said, single end strands of double-stranded DNA. And so the 5' fragment will be sequenced from the positive end, the 3' fragment from the negative end. And we can predict the whole fragment by extending in either direction from the 5' or 3' end. But this is kind of a better visualization of it, where you have your antibody binding to this particular region. This is probably going to be some kind of peak region. And you have, these are your entire reads, but the green and red parts are the parts that you've sequenced. So you're really only sequencing, the ends, and the part where the antibody is actually binding is actually not represented in your data set as strongly. And so by assessing the fragment length, we can actually get a full picture of what this enrichment region looks like. And so this is another representation of how it might look, where the green are going to be your negative strand, the red are your positive strand. And so you'll have a buildup of reads here, a buildup of reads here, but what your actual enrichment looks like is going to be here. So we have algorithmic ways to understand what this means and how we can eventually get here. This is a cartoon that kind of displays this. So what cross correlation is doing is it's basically looking at the correlation of overlap, the correlation of read signal in the positive strand as you kind of shift it along this axis and correlate it with the negative strand. So when you start out, there's not much correlation. Then as you shift these reads, the point at which you have your fragment length, you will get the maximal correlation, and that allows you to predict what the fragment length generally is. And so this is what the output will look like when you actually do this correlation. You usually have this peak right here, which is about the length of your fragment. And this is typically going to be representing the extent to which you have artifacts in your chip. These are low complexity peaks that are essentially just usually sometimes single reads either stacked on top of each other or the absence of a read there. If it's a place that can't be mapped, will also produce this. But because it's just a bunch of reads stacked on top of each other, the correlation is just going to be the length of your fragment, or the length of your sequencing read as opposed to the length of your fragment. Whereas this peak that is a little bit later on is typically going to be your fragment length, where there's the highest level of correlation between the positive and negative strand. We can use the plotcc function from ChIPQC directly on this ChIPQC object to get these cross correlation plots for our samples. And if we use the facet by argument, we can, by sample, it will plot each of our BAM files. And so, you can see we have a wide range of shapes here, where we always have this artifact peak, and you can see is pretty prominent. For our 4 chips, our fragment length peak is actually usually the same or higher than this fragment, so that's good. And then you can see the inputs, which should be less peaky, have a higher artifact peak and a much lower fragment length peak, which makes sense, because if you don't have nice enrichment, you're generally not going to get the high levels of correlation that are caused by shifting peaks. We can also include metadata as a data frame in this plot.cc function, and that'll be helpful because we can try and facet by different things and help our visualizations to understand what's actually going on. But here we're creating a data frame that has our sample name, the tissue that it came from, and whether it's an IP or an input. And so with this metadata, we can include this in our plot.cc function. So we add the metadata, and then we can map on things like colors to specific columns of that metadata. So one of our columns was antibody, so we're going to color by that column. And then we can also facet by tissue, which was the CH12 versus MEL. And we get this nice plot where we can clearly visualize that you know, our chips clearly have much better cross correlation at the fragment length than our inputs, which is good and what you want to see. All plots in ChIPQC are just actually built within ggplot under the hood, so you can update your plot like any ggplot object. So here we're using the plot.cc function to build that plot that we just had, but we can simply add things like themes or titles in the same way that you would to any ggplot object. And so blacklists and SSD are more metrics that are present in this ChIPQC sample object that we can use to assess quality. And so just to go back to blacklist a little bit, this is kind of visually what it might look like. This is the bed file that this is an older build that we brought in from potentially the ENCODE website. We put it into IGB, so this is the region where someone else has curated the blacklists and determined this is a region where typically you get blacklisted ultra high signal. And you can see, I mean, this is a different experiment, but you can see that, you know, you have these things that are just incredible amounts of signal. If you were to zoom all the way out, you'd probably, you typically see in your IGV, I don't know, a handful of giant peaks. And when you zoom in, they're just like your peaks are going to just be really, really tiny compared to this. And so this is both going to, this can potentially throw off. Most peak callers can deal with these pretty well, but this can confound peak calling and fragment length estimation. So it's important to hopefully catch them with the blacklisted bed files and exclude them from most of your analysis. Just to show what we meant by this artifact peak at the beginning. So this was just a particular sample set where we're looking at, we've separated the reads into those that fall in peaks, those that fall into duplicates, the blue, and then reads that are aligned to blacklisted regions. And you can see that obviously in our peak region, we're getting this nice peak at our fragment length, whereas in a little bit with duplicates and certainly with blacklist, you can see both duplicates and blacklist are contributing to this artifact peak. And so again, this is just to show that this first peak here can be a rough estimation of your blacklist compared to your enrichments. SSD, standardized standard deviation, is another measure that's sensitive to blacklisted artifacts. SSD is essentially measuring the standard deviation across the entire genome. And so this can both be a reflection of enrichments, like good enrichments, biological enrichments, and peaks, but generally blacklisted regions will also dominate this measure as well. And so you can see here that in this particular figure, the last three samples are getting rid of blacklisted regions, and here you have blacklist. You can see SSD goes way down without blacklist, but you can also see that SSD, after you remove blacklist, can also correlate with chips where your inputs have low levels of SSD, because generally they're just going to be flat, so you're not going to have much deviation of your signal, whereas as you look at your actual chips, you can actually get some significant increase in SSD if you have really efficient chips. So the plot SSD function can, from the ChIPQC package, will take your ChIPQC objects and plot your SSD. And if you have a blacklist, it will show pre and post blacklist. So you can see the extent to which, the big extent to which, once you filter out blacklists, you reduce SSD pretty substantially. So this, SSD of each individual sample, sorry, it's loud, of each individual sample will not be super informative, but both compared to other samples and most especially compared to pre and post blacklisting, can be quite informative. So you can see the extent to which blacklist is contributing to SSD for each sample and then compare that to the others. You can also just zoom in to the particular region of your samples without blacklist, because obviously if there's a lot of blacklisted driven SSD, it's hard to sort of assess what your samples look like without blacklists. So you can do that. Here you can see actually the input has a higher level of SSD. If you go back to our cross-correlation plots, you can see this particular input had a really big blacklisted peak. And so sort of what this means is that there's probably, once you got rid of the blacklist, there's actually some artifactual regions that remain that weren't in the previously curated blacklist regions. And that's probably fine, but it's potentially something to be aware of if something looks weird further down the line. And so just a note on library complexity. So library complexity is another source of noise in ChIPseq, and this often can come from over-amplification of your library, and that usually results from you having a really low input of DNA to start with before your PCR step in library prep. And this is what a low complexity read might look like, where you just have a pile up of a duplicated read as opposed to a typical ChIPseq read where you have, reads from multiple fragments that clearly are coming from different places that eventually pile up to form a peak. And so we should and can easily compare our duplication rates across samples to identify if any particular sample was overamplified compared to the others. And we can use the flag tag counts function on our ChIPQC objects. This is again our list of ChIPQC sample objects. And if we do that, we get a data frame we can access and look at the duplicated over the map to get our rate of duplication per mapped reads. And you can see we have between 7 and 15% duplication amongst our alignments, which isn't super high. That's not too bad. But again, it's something that you kind of want to be aware of, especially comparing across sample if one sample eventually looks strange. And as with any of these metrics, there's, obviously there's been lots and lots of chips done, and ENCODE usually does a pretty good job of giving you reasonable thresholds or at least points at which you should be concerned. So you can really scour the ENCODE website and try and understand like what is a reasonable duplication rate and stuff like that. But So we can also understand just with, using ChIPQC, we can easily understand where our distribution of reads across gene features using the plot regi function. And so this is just a really easy way to understand whether our reads are enriching in certain parts of the genome. And so this is the output. So you're just using, again, the list of your ChIPQC sample objects, and you get this nice plot that divides the genome into different features. And you can see a nice enrichment, which is what we'd expect for MYC. We know it, among other things, binds to promoters, and so we see this enrichment here of promoters in our chips over our input. You'll notice that there is still a little bit of an enrichment. Like if this was scaled differently, you'd actually think that there was an enrichment in your input in your promoters. And this is just, again, going back to the fact that typically when you sonicate, you're going to slightly enrich for more open regions of the genome anyway. And that's why you really want an input, because you want to make sure that this enrichment that's coming from your chip is actually greater than the enrichment that's coming from your input. And so we'll now move on to peak calling. And so peak calling is essentially, we looked at our bigwig and we saw that there appears to be regions where there's enrichment of our signal over input, but we can understand statistically whether that's actually true. And there's various peak callers that work in different ways. By far the most popular is max2. Generally the way that I'll work is I'll start with max2, and then again, different peak callers have different strengths, whether you have sharp peaks or broad peaks, and all things have trade-offs. Generally the broad peak callers might be a little bit more I guess less aggressive with how they call peaks. So you'll get more coverage across the genome of peaks, but you might actually pick up your broad regions, but you might call peaks in areas that actually don't look very peaky. And so oftentimes it's an assessment of just actually calling peaks with your peak caller and then looking in IGV and seeing if it feels like, if it looks like it's actually catching the things that look like peaks by eye. And so it can be a little bit of optimization, and you'll get a handle for which peak callers you like for certain chips. But generally max2 does pretty well out of the box. And so that's usually where we start and where people usually start. But max2 calls peaks in a few simple steps. So it predicts fragment length. It then shifts reads to the center of the predicted fragment, and then scans the genome and looks for enriched regions compared to control samples using a count-based statistic. So there's no R package for max2. Well, actually it's coming, but it's just about to be released, and we'll test it and hopefully it will work really well. But generally when you're using max2, at least right now, you're using it on the command line, or you're using R to interact with the command line, which is how we'll do it. You can install it pretty easily using the Anaconda package repository. If you have a Windows machine. And what Anaconda is, and I guess you used this for Salmon for the RNA-seq lecture yesterday, but it's just a big collection of version controlled packages that can easily be installed through the Conda package management system, which is nice because you can create environments and hold for example, different versions of different packages in different environments. So you can jump in and out of these environments and use specific collections or specific versions of packages. But at the BRC, there was a package developed called Herper, which again you used yesterday, but we'll go over quickly again today. That package was spearheaded by Tom and Matt. And what this package does is it sort of allows you to interact with that Anaconda package system, staying within R, both getting the software from Anaconda and also using these environments in a way that typically is not as easy to do in R as it is in the command line. This is a bioconductor package, so we can install it with Biosq Manager. We load it to our environment, our current R session. And with the install conda tools function, we can identify the package we want to install, which will be max2, and also the environment to install that package into. And so what Herper is going to do is it will go get a minimal version of Conda, which is very lightweight and just includes the sort of bare bones necessary packages to make it work. It will then create this new environment called ChIPseq Analysis, and it will install max2 into that environment. You can also, if you already had ChIPseq Analysis environment existed and you want to put max2 into it, or put another package after you install max2 into this environment, you can use the update and function to do that. So max2, if you're using on the command line, this is what the command would look like. So we're using the call peak function within max2. The -t flag is going to be, or argument is going to be our chip BAM file. This is the name that will be appended to the output files, and this is the directory we want those files to go to. And then the -c argument will be, hopefully, but highly recommended, you have an input file that max2 will compare this chip to. And you can do it without an input file, but it generally, again, it first doesn't work nearly as well, and also it gives you certainly less confident peak calls. So if we want to run this in R using both the R environment and using Herper, we can easily do this, and we can run it in our specific environment. So we've already installed max2 into the ChIPQC analysis environment, so we can use the with conda m function to use this environment, and then use this, and then it will run this function within this environment. And so what it's going to do is we're just essentially building this function call using the system2 function, which is either the system or system2 are two functions that allow you to run command line functions from RStudio, or from R in general. So we just essentially build these arguments, run it, and it will do our peak call. And max2 will just automatically generate a number of files and put them into the directory that you specified in the peak, or in the function call. And so there's a couple of files that get output, one of which is this, it's a tab separated file that they label as XLS, but you can open it in Excel. But this will have your peaks, and also at the top of the file, it has some really important information about your function call, which is nice because oftentimes if you're running things from the command line, it can be harder to document because you don't have a script, but you can have a script. You often will just run it. And unless you, for example, copy and paste that line of code into somewhere else and then save that file, you can easily lose track of exactly what you did. But this XLS file from max2 has some commented lines at the top, which have some nice information. Most importantly, it has your actual line of code that you ran, which has your BAM file and has your input file. It also specifies those files for you. So this is just nice to remember that for max2 at least, you will have some pretty good documentation as long as you have the output. Later on in this file, if you don't read in these commented lines, you just have a nice what looks like almost a bed file where you have your the genomics position. So these are places where peaks were where enrichment was found and peaks were actually called. And there's some statistics such as p-value, fold enrichment, where the actual highest point of the enrichment was. And this log 10 p-value and fold enrichment are measures of significance, meaning the higher the value, the more, usually the larger the larger the number, the larger the enrichment there was compared to input. And so this can be, if you're interested in ranking your peaks, that these can be good metrics to measure them or to rank them by. So we'll read in this file, this peaks.xls file, omit the commented lines. And now we have this in a table. We can create a granges object, which, if you remember, a granges object is a nice bioconductor object that can easily be converted to bed files to look at IGV, but also we can easily interact with them with many bioconductor functions. And they also print out nicely to the screen where you can see the exact range of where your peak actually is. So using the genomic ranges package, we use the granges constructor function, which we need to take the seq name or add the seq names, which is going to be the CHR or the column within this data frame that has the column name CHR, and then we build the ranges with that just takes the start and end, and in the end we have this granges object. And now that we have this granges, we can use there's lots and lots of different functions that can be used on them, and accessor functions that allow you to pull out certain parts of this granges. So we can just get the seq names with the seq names function. We can just pull out the ranges with the ranges function. We can also add metadata to this. So before, all we had was the sort of necessary parts of the granges, which are the seq names, ranges, and strand, but you can easily add metadata with the mcalls function. So we'll take the summit and the fold enrichment columns from this max2 output data frame and add them as metadata to our granges object. And you can see the output here as we nicely have a row for each peak and also the corresponding summit and the fold enrichment. One other note, which might be a little easier if you want, is also output from max2 is this narrow peak file, which is essentially just a specialized bed file. And you can actually look at these in IGV. You can open these in IGV and they'll work. But you can also, and this is a recent addition to our track layer, but you can actually just import these directly using the import function, as long as you specify format equals narrow peak. And then this will give you a granges that has all of and it'll add metadata columns for the extra columns that are in this narrow peak file, which you can't see them down here, but it specifies the peak and has some of these metrics that tell you how enriched the peak actually is. And these are the scores that you see. If you open a narrow peak file, the different peaks will be different colors, which is kind of nice, and it will indicate whether how highly enriched that peak is. So this is essentially the same output, not the same because it has different columns, but it's a similar output as this. So there's just two different methods to get to a similar object that can be used for downstream processing. So as we've talked about, blacklists are an important consideration. And so they're important to be aware of for QC, but they're not important for any sort of downstream processing and sometimes can get in the way. So before we move on with our analysis, we'll get rid of them. So we use our track layer to import the bed file that contains the blacklisted regions. And so this object here will be a granges of our regions. And then since we have a G ranges with our peaks and a G range as a blacklist, we can use this over function, which is a nice operator specifically for G ranges, where it will go through every line of the first G ranges and give you a true false value. So it'll return a vector of true false values for every peak or for every line in your first G ranges, telling you whether it overlaps any peak or any range in this second G ranges. And so since we want the peaks that don't overlap our blacklist, we'll get a logical saying whether a peak overlaps or not, and then we'll take the opposite of that, and we'll filter our G ranges or subset it based on those that don't overlap blacklist. So now we have, we've done our alignment, we've called peaks, we have a nice set of peaks that aren't in blacklists for this particular sample. And one thing you might do straight away, which is pretty easy in Bioconductor, is to annotate those peaks. And you can annotate those peaks with things like types of genomic regions. You can annotate them with genes. And there's different ways to annotate, especially with genes. The simplest way is just to annotate a peak with a gene if they overlap that gene, or you want to find the closest gene, and you can set sort of minimum distances. And this is obviously important because, you know, you can scan throughout IGV and try and understand where your peaks may or may not fall. You can go to your gene of interest, that your one gene that you know and that you're interested in, and see whether there's a peak there. But obviously the benefit of whole genome or of sequencing experiments and next generation sequencing is being able to assess things at a genome-wide scale. And so by annotating all your peaks and doing some of the downstream steps that we'll talk about later on today, you can really start to understand and try and identify functions of functional impacts of your chip that you may not have predicted without looking at the whole genome and looking at genes that you already know might be important in your particular experiment. And so a useful package for annotation of peaks is the ChIPseq package. And we'll combine this with a predefined annotation of each particular genome build housed in this TXDB object, which is a Bioconductor object for your particular genome. And we will also use this annotation database that is that nicely interacts with all these Bioconductor objects, including this TXDB. We'll load ChIPseq, and once we have all those libraries loaded, we can run this annotate peak function, which is a ChIPseq function, which just takes the G ranges of our peaks. You can define what you want your TSS region to be. So how aggressive do you want to be with what you actually define as a promoter region, your TXDB object, and also your annotation, which will allow you to map gene names onto certain gene IDs and gene regions. And so you run this function, and what you get if you look at the class of this, what the result of this function is this CS anno object, which again is just an object that's specific to this package. But as we'll see, there's some nice functions that can be used directly on that object to get some nice visualizations and information. And so right out of the box, what this object looks like is it gives you this nice table that tells you how many of your peaks actually were annotated, and the frequency with which your peaks overlap with certain genomic features. And so each peak, at least in the ChIPseq function, is only annotated with one feature. And so this says that 41% of your peaks fell within promoter regions, which, and 21% were in these distal intergenic regions, which maybe you could argue would be enhancers based on sort of known functions of MIC. Enhancers and promoters are places we know it goes. And so that's interesting that a large proportion of the peaks fell within those types of genomic regions. The CS-ANO object can easily be coerced into granges with the as-granges function, or you can just use the as.dataframe directly on these objects. And this is nice because it will give you something you know what it looks like. You can easily interact with it. And here, for example, the granges version of it, you have the peak and the metadata that we had appended to it before, so it just takes the granges that you put in, and then it appends a lot of interesting information, such as the annotation. So where did it fall? Is it a promoter, distal, intergenic, intron? And then it has the annotation of the closest gene, and you have your ID, the symbol, and also it tells you somewhere how far from the TSS you were. So this is related to how we defined promoters before. We did plus or minus 1,000, I believe. So this one directly overlaps the TSS. This one is 50 base pairs away. And so you can use, this is just a grange, so you can use all this information and filter it however you like. You can easily just get some nice visualizations directly with the CS-ANO object. You can use plot ANO bar, and there's also a pie chart version that directly gives you a sideways bar chart, a flip bar chart, showing visually the percentage of reads that fall into these different regions. So as we saw before, 41% were in promoters, and this just shows it nice and clearly. And this is also nice because you can do this with multiple samples, multiple chips, and stack them on top of each other and easily see how different samples compare to each other. This is another one where it tells you the percentage of peaks that fall within certain distances of TSSs. So, here we can see even, about on each side, maybe 30% total of our peaks fall within 1 KB of transcription start sites, which again makes sense for MIC. You can also easily do an upset plot, which will tell you in a succinct way whether there's overlap between different annotations. And so, yeah, the ChIPseq is just a nice package that allows you to really quickly, with just your granges, understand very quickly, like at a high level, some things that, or some places or annotations that in genes that your peaks are falling near. And we'll move in this afternoon to try and understand how we can take that information and maybe get some statistical representation of overrepresentation or functional enrichments and stuff like that. So again, there's more exercises for this lecture. Exercises and answers can be found here. I believe, let's see, yeah, so we're done now until 1 o'clock, where we'll do functional enrichment and motifs. If anyone has questions now, certainly ask questions in the chat. I'll be around, so you can also just speak up and ask me directly if you like. But I'll probably, if anyone has any questions right now, I can answer them and try and even, working them in our studio. We have a question from Sadie. What certainty do you have for the second factor of proximity, but not overlap in assigning gene, or any guidelines, and how aggressive to be with TSS? Yeah, we'll cover a little bit this after, because there's definitely different ways to annotate where you're not just looking at the closest gene, because obviously, especially if you're looking at something like an enhancer bound factor, then enhancers can regulate multiple genes, and so there are different ways to annotate. As with anything in terms of this, like all of this is just hypothesis and prediction driving, so nothing is obviously for sure. in terms of TSS bound, in my personal experience, generally TSS bound factors are going to bind pretty close to the TSS, I think. And you can, I think the best way is to just look at papers that do this and see what people typically use. I think it's pretty common to go 500 base pairs downstream and maybe 1 KB upstream. But that's also, like, these are things that you can easily play with these functions and see how it, like, I guess in the end, I would advise you just kind of like play with your data. If you have a transcription bound factor and you keep running this ChIPseq function and you reduce your TSS window and you know, what might happen is you actually get, you know, some will drop off, it'll change your annotations, and it might actually be interesting in a way, because then you're getting the genes that you have a higher confidence that your factor is actually binding the promoter. So I think it's something that you just kind of both read about and play with, but again, it can change your output. So it is important to consider. But again, I think TSS bound factors usually are binding pretty close, so you can be pretty safe with probably 1 KB around, and that'll probably catch most genes or all genes that your factor is binding to. Yeah, hopefully that helps. All right, so obviously we're still dealing with the workflow for ChIPseq analysis. This morning we worked through some Fastq quality control, then some quality control of our BAM files and our alignments, along with peak calling and some early annotation of peaks. So now we're going to take those annotations and try and see if we can understand anything that's going on with our chip and make some, not necessarily conclusions, but, you know, but hopefully get some hypothesis generating analyses that will then allow us to go on and do some interesting experiments. So since we were doing this today, we don't really need to go over the data, but I guess we'll start using data from all 4 samples. But we have two replicates of MIC ChIPseq from the MEL cell line. This is the mouse cell line, and 2 replicates from the CH12 cell line. And these can all be found and downloaded with these links here. Also, the peaks files are actually just going to be in the data directory if you downloaded the materials for the course today. So they'll be in a subdirectory called peaks, and within that will be the tab separated files you can open in Excel that are the direct output of max2, which is the peak caller. And so we had done this earlier for one of these files where we, using the genomic ranges package, we use base R to read in this tab separated file, and then we construct our G ranges from this table, which we read in as a data frame to get the chromosome, the start and the end, and also we attach the metadata, just like we did this morning, and attach the summit and fold enrichment columns. And so here we have our constructed G ranges object, which are the peaks for this particular MEL1 replicate in G ranges format. And again, this is just a format that is used throughout Bioconductor, and we have lots of ways to interact with it. So it's very, very useful. And so since transcription factors, you know, obviously are trying to regulate transcription of target genes, and we've already annotated our peaks with the ChIPseq package to associate our peaks representing binding events to their overlapping or closest MM10 genes, then we can go forward with this. And again, just to, it's a good question earlier, you know, how confident are we in these annotations? It depends, you know, we're going to use promoter-based annotations, and so those you're usually a little more sure, but as with anything in science, you know, this is a hypothesis and it obviously has to be tested, but annotations can be good and some can not be so good. So it's really just a place to try and explore your data and see if you can think of some interesting questions to move forward with. But So similar to what we did earlier, we'll use this annotate peak function from ChIPseq, which will take our G ranges. We'll define what we want to define, or what we want to call a TSS, and this will be the promoter category in the annotated G ranges. Our TXDB object, which will indicate where in the genome genes fall. This is the N10 genome, and then our annotation database, which will help us map gene symbols to gene IDs. So it goes through all these steps. This is what's printed out to the screen. And then at the end, we get this CS anno object, which we called peak anno. And this can be converted to a G ranges or a data frame very easily. And here we're showing the data frame, which has the columns of our G ranges, including the metadata, and then these additional columns that show what type of annotation it falls in, whether it's promoter or intron or exon, and then information about the closest gene that it was annotated with, including the gene symbol. And so this is obviously nice, and it's interesting that if certain genes pop up, but we want to move forward and try and look for things that aren't obvious on the surface. And that's typically what you're doing with gene set enrichment. And so gene set enrichment is just simply taking a list of genes. It's essentially very similar to what you guys did for RNA-seq when you were defining your gene set by differentially expressed genes. Here, we're just defining our gene set by those that are annotated by our peaks. And this is just trying to answer the question of whether our transcription factor or epigenetic modifications are acting on a specific set of genes that's usually been curated by someone else. And there's lots of nicely curated gene sets. I'm sure some of them you've interacted with before, such as gene ontology. KEGG is another popular one. Another one we'll use today is MSigDB. We'll talk about that a little bit later on, but this is lists that are both computational and experimentally derived from the Broad Institute. And so, like I said, gene set enrichment, or proteins can obviously be binding anywhere in the genome, not just in promoters. But in this case, since we know, and we determined earlier on that a large proportion of our genes that MYC is binding near is in the promoters, we're going to use those promoter-bound genes, because those are the ones we're pretty sure if it's binding to the promoter, there's a good chance that it's regulating that gene. We're not going to go and interact directly with these databases. There's lots of nice R and bioconductor libraries and packages that allow us to access them. One of those is the cluster profiler package. So this is a package we started to use a little bit more in the core recently. And it both does the enrichment for you and also has some nice functions that allow you to directly access some of the databases like gene ontology and KEGG. And as we'll see in a little bit, you can also get your own custom gene sets and test your gene list against anything, as long as you have a set of genes. This is a pretty well documented package. There's this sort of book. The author of this package, this person here, has a lot of bioconductor packages, so their documentation is usually pretty good. But if you do end up using this package and you like it and you're interested in going further, there's lots of different functions and types of visualizations that you can do with this package. So there's a lot to explore there if you're interested. So like I said, for this particular analysis, we're going to focus on promoter-bound peaks, and we know that there is a column in our G ranges that specifies whether our peak is in the promoter of the gene that it was annotated with. As we've seen before with G ranges, you can simply just subset them based on columns in their metadata. So we'll subset this G ranges based on those, by filtering for those rows or those entries that have promoter in their annotation column. And then we then want to get just the gene ID. So instead of having a G ranges, we just want a vector, a character vector, a numeric vector of the actual gene IDs. And then we're going to use the unique function, which is a base R function, which will get rid of any duplicate gene IDs. And this is important because you can have multiple peaks that are annotated to the same gene. And so in that case, in this table, you'll have, or the result in this vector is you'll have multiple entries of one gene potentially, and that can throw off your gene set enrichment test. And so it's important you just want a list of all the genes that are annotated and make sure they're only listed once. And so then another important step for gene set enrichment is to define the universe of genes that are possible in your gene list and gene set. So since we use this TXDB object to annotate our peaks, this is essentially the only genes that are in this that are in this TXDB object can show up in our annotation. So we will extract all of the genes from this, just first using the genes accessor, which will pull out this G ranges at the gene level, and then we can isolate the gene IDs by just using the $sign and accessing this column here. And so once we have our gene lists and our universe of genes in the same format, we can use them in the enrich_go function, which is a cluster profiler function that is specific to gene ontology. And like I said, there's other functions. There's another function that's enrich_keg, and there's, but this one again is specific for gene ontology. And gene ontology is just a very nice broad set of gene lists or gene sets. And one good thing about them is there's been care taken to have, to understand the connections between the two and relationships between certain gene lists based on the commonality of the genes that they contain. So we'll load cluster profiler, we'll load the annotation database, and this enrich_go function just takes so this is the vector of the genes that we're interested in. This is our universe, so all the genes that possibly could have been in this list, our annotation database, and then this ONT argument for ontology. It can take one of four things: BP for biological processes, MF for molecular functions, CC for cellular compartment, or you can include all three. And these are the three main types of gene sets that the Gene Ontology Consortium fits their sets into. Here we're going to just use biological processes. So you run this function, and the result is this enrich result instance, which again is just another cluster profiler specific object that contains a lot of the information that comes out of this analysis. It can easily be just transformed, or you can access it as a data frame just using the data.frame function. And you can see you get this nice output that lists ranked by p-value the top lists that were enriched from the gene sets that you chose. In this case, we did gene ontology biological process. And you get your p-values. You even get the gene IDs from your list that overlap with the particular gene set in that row. And you can convert these to gene symbols, sort of as we've done in this course. And so one nice thing about cluster profiler is there's pretty good ways to directly visualize it. And that's through this other package called enrichplot, or at least this particular network plot that we're going to show. And this was written by the same author that wrote Cluster Profiler, which I assume is why they interact so nicely. But you can just take the output, so not even the data frame, you just take the enriched result output, and apply first this pairwise term stem function, which is just going to add information to this object that will indicate sort of relationships between similarity relationships between terms in your list based on how common overlap of genes in the gene set. And so you have your updated object, and then we use this emap plot function. You can tell it how many of the top significantly enriched lists or sets you want to show. And we get this nice network plot, which especially for things like gene ontology, where you get a lot of terms that seem very similar, like RNA splicing via transesterification, or RNA splicing, which is probably the parent one, and then, you know, different mRNA processing. But this way, things are grouped together in a way that you can just look and sort of see, oh, here we have some sort of mRNA, RNA sort of splicing or processing group, and then this is more tRNA and rRNA and nuclear non-coding RNA. So you sort of get two large groups instead of this big list that is kind of hard to parse through. And for gene ontology, especially where a lot of the gene lists overlap, this works pretty well to help you really just get a higher level and digest these lists a little bit better. So next we're going to use another cluster profiler function, Enricher, which works a lot like EnrichGo, but this is what you'd use if you want to test your gene lists against custom gene sets that we can either import from as GMT files, or GMT lists if we have them, or we can get them from somewhere else. Here we're gonna get our list from the MSIG database, which again is this database. Information is here, I believe. Yeah, so it's from Broad, and they've basically done their own curation, both with sets that are determined computationally, such as here, or data that's been mined from publications on PubMed or expertly curated. So it's just sort of this larger bucket of gene lists that generally can be a little more targeted than the ones in gene ontology. You know, for example, some of these experimentally determined lists might be knockout of a particular factor. So if, you know, if your peaks are enriched in a gene set when this thing was knocked out, this factor was knocked out, then maybe, you know, your transcription factor you chip for might be related to that thing because its gene list, that protein because its gene list was enriched in your annotated peaks. So it's kind of just a nice broad set of gene lists. And there's also an R package, MSIG DBR, that allows you to access these lists. So you could just go to that link, this link here, and download the GMTs yourself and read them in. But recently this MSIG DBR package was developed, which just allows you to get them much easier. And so you can use this MSIG DBR collections function. You just run it with no arguments, and it basically spits out a tibble, which is, if you remember from the tidyverse class, is very similar to a data frame and how you can interact with it. But it just has a column, and this is a much longer table than this, but this has a column with the gene set category and the subcategory, and the number of gene sets that are contained in that category. So for example, KEG is actually included as in the C2 category as a subcategory. And I believe all the gene ontology categories are actually in here too. So if you really liked interacting with MSIG, you could actually just do your gene ontology with the enricher function by just specifying the correct subcategory. But anyway, you can look through this tibble and get your categories and subcategories, and that's what's going to be used to actually get your gene lists, which is done with this MSIG DBR function. So what this function requires is the species, and I think there's an MSIG DBR species function similar to this one that tells you what species they have available and what you actually would put in here. But we're going to do mouse. Our category is H, which H, if you look for that in the table from the previous slide will be hallmark gene sets. And these are just kind of their broadest gene sets. And there's not that many of them, so it's quick, which is why I did it here, but it also is usually a good place to start because there are some pretty broad gene sets that can maybe point you in a certain direction, then you can get more specific if you want. And we're gonna look at all the hallmarks, so we'll leave the subcategory as null. So we run this function, we pull out the hallmark gene sets, and then so for enricher, we basically need, which enricher is the cluster profiler function. We need a data frame where the first column contains the gene sets, and the second column are the genes that are in that gene set. So you'll have multiple entries for each gene set based on how many genes are in that set. And so here we're going to subset this. I believe this is a tibble that comes. Yeah, it's a tibble that comes out. So we'll subset this tibble because there's multiple columns in here that we don't want. We'll subset it based on just the gene set name and the corresponding entrees IDs. This is what it looks like. And so we can use that data frame in this term to gene argument within the enricher function. So it's very similar to enrich Go, where we have our gene list, our universe, and then instead of specifying a gene ontology, we're going to use this term to gene argument and just put in our custom gene lists or gene sets. And so we run this enricher function. The output, which we've called hallmark, is going to be the same exact type of enrich result object that was the output of the enrich Go function. And if you remember, we can easily just turn that into a data frame and we get our list of enriched peaks or enriched gene sets, sorry, from our annotated genes from our peaks. And this is a nice sanity check. You can see there's a MIC hallmark gene set, and there's apparently 2 versions of it, and those 2 come up in our top list of enriched gene sets. And so, hopefully if you expand this list a bit, there might be some interesting things. I don't know exactly if E2F is a known cofactor of MIC, but potentially you could identify E2F as a cofactor, and that would be an interesting sort of conclusion from this analysis right here. But again, there's tons and tons of MSIG gene sets, and so it's something that you can just kind of play with and cycle through and try yourself with your own lists. And so another popular gene set enrichment package is GoSeq, and I think you guys used this a little bit yesterday with RNA-seq when you were looking at your differential expression gene lists, but we can use it in the same exact way here and very similar to how we use cluster profiler. There's a couple sort of details that are different. Namely for GoSeq, instead of having separate, like just a vector of our gene list, we need a named vector of all genes in the universe with 1s or 0s representing whether that gene was annotated with the transcription factor bound peak or not from our chip. And so we can easily do this by using this in operator, which will just tell you whether the second, any entry from the second list or vector is, well, I guess it'll go through this first vector and then tell you whether that is in this second one at all, with a true or false for each element of the first vector. And we can convert this logical vector that comes out from this operation to an integer by just using the asInteger function. And then we'll name them just with the genes that we evaluated. So the output will be, again, a named vector where each element is named with a gene, and then you'll have an indication with the 1 whether that gene was actually in your set. And if you remember, one nice thing about GoSeq is that it actually tries to incorporate gene length bias. So this is especially true for, or relevant for RNA-seq, where because larger genes have more reads in them, they're more likely to be called significant because they'll have a lower standard deviation relative to their counts. And this can be relevant, I suppose, for our ChIPseq too, because a bigger gene might be more likely to have more peaks near it if it's quite large. But it is still a good thing to do, necessary if we're going to use GoSeq. So we do this with this null p function. And all this function takes is that named or vector with the ones or zeros that we just made in the previous slide, the genome, the type of ID you have, and then it can output this plot that can sometimes be helpful, but we're not going to output it today. I think you saw that yesterday. So you run this function, you get your null p object, and this is what's going to be used for GoSeq. So the GoSeq function takes that output, and then the gene2cat category is the same essentially as the term to gene argument that we used for the enricher function from Cluster Profiler. It just needs a data frame, a tibble. I realized won't work, and so you just need to convert this tibble that you got from the MSig DBR package to a data frame, and you get a data frame as an output from GoSeq, and you'll see with very similar p-values, we actually get the same results, because here we're again using the hallmark gene set, because this is the gene sets that we extracted from the MSig database. So this is sort of just a nice sanity check that sometimes it can have an effect, but it's nice when common packages are robust enough that they give you similar results. And so this is sort of getting at Sadie's question from earlier a little bit, where so far we've just annotated with closest genes, and that can obviously be limiting because oftentimes peaks or transcription factors can bind to a certain place and regulate, if there's some kind of TAD there, it can regulate any gene within that regulatory region, or maybe it skips over the closest and regulates the next one. So, again, all these things we don't know until we experimentally test it, but in terms of trying to generate hypotheses, it might be better to be more inclusive with the genes that we attribute to each peak. And in this case, there's this great tool set which allows you to incorporate distal peaks and sort of set rules that tell you which peaks to include. And there's some pretty good explanations on their website about the different statistical tests and kind of how they actually annotate peaks to genes. But essentially what they do is they define a regulatory region for each individual gene, and then essentially compare the proportion of peaks that map to that regulatory region to the proportion of the genome occupied by all the regulatory regions in that gene set. And if those regulatory regions are overrepresented compared to those you'd expect, you'll get an enrichment score. So it's just basically a way, like you'll obviously get a longer gene list, and you don't even have to use the great tools for the annotation. You can just use great to get your gene set and then go use that in Cluster Profiler if you want. So it can just be a way to annotate, and then use other tools to enrich. But there is a user interface you can use, but there's also a nice bioconductor R package. that we're going to use today. So we'll load that in. So the first function you'll probably interact with the great tool is the submit great job function, which will return a great job object. What this function takes is it just takes a GRanges. So these are our peaks. It takes the species. They recently updated to version 4. There are some, so version 4 is nice because it includes HG38, but it doesn't have quite all of the gene lists that version 3 did quite yet. So we haven't changed this yet, but right now this is version 3, but you can update this to version 4 by just putting in 4. This is if you start using this a lot, this can be a helpful parameter. For some reason, I assume to limit the number of requests that hit their server, they limit the number of, or they limit the time in between these function calls. And I think this is like 2 minutes or something by default, but you can just change it to 1 or 0, and then you can keep submitting things. I think they just by default want to limit the frequency with which people submit jobs. So we have our peak set. We do the submit great job function. It goes off and annotates the peaks, and then we can see the available categories. So great, in addition to annotating peaks, like I said, actually does the gene set enrichment for you based on their own algorithms. And we can see the possible categories with the available categories functions. We have gene ontology. We have some curated lists for gene expression, certainly, or some types of gene families, regulatory motifs, pathway data. We're gonna look within regulatory motifs. And so you can get specific enrichment tables within these different lists by using the get enrichment tables function, which just takes the great job output object and takes the category. And so, of course, it gets even more granular, and you can look at the names of your table. And there's now 2 different subgroups within the regulatory motifs. There's predicted promoter motifs and mRNA motifs. And you can see some of these. This is from the ChIPseq database. So a lot of this stuff kind of overlaps because everyone's using a lot of the same gene sets. So then you can take your great result table, and this is just a list with the different subgroups within it from regulatory motifs. And so we're going to subset this list based on this ChIPseq predicted promoter motifs category. And you get a nice data frame out from this. If we look at the first 4 rows, you can see that we have, so this is again promoter motifs, and you can see that we've overlapped with MIC is here, so we've overlapped our MIC chip with MIC motifs, which is a good thing. That makes us feel good. But there's also other interesting things that pop up, and again, I don't know if these are known cofactors, but this is sort of where you can very easily start to generate hypotheses and test these things. So that's exciting and fun. So this is, I guess, a good segue into our next section, which is motif enrichment. So this is just based on the fact that transcription factors often bind to specific sequences throughout the genome. And these have been experimentally determined, usually by just simply doing a chip and then looking for enriched sequences under those peaks. And so a common practice is to take your transcription factor, look at your peak, see what sequences are enriched, usually some sort of de novo motif enrichment. So you're identifying the most enriched sequences and then comparing those to known motifs And that can be another way to try and identify cofactors or other functional consequences of your chip. And so there are some nice R and bioconductor packages that allow you to do this, but they can be pretty time consuming and memory intensive. There's a nice user interface called Memechip that does de novo motifs, and we can easily prepare the input for Memechip using R and using bioconductor, and so that's what we're going to do today. And so what Memechip requires is a FASTA file of all your sequences under the peaks that you're interested in, and then it'll just look in those sequences and look for motifs. And so, as we've done before, we can use the BS genome package. Remember, this is what we used to get our FASTA for our alignment. But we'll load in BS genome, load in the MM10 genome specifically. And another thing about transcription factor motifs in general and Memechip specifically is that it works best if you're looking at usually about 100 base pair region, but also it's important that it's near the summit of the peak. So, obviously the summit of the peak is where we think the transcription factor is binding the strongest, and so this is most likely where a motif would be for it to, if it's driving its binding. So we're going to make a G ranges. So if you remember, one of our metadata columns of this G ranges was the summit. So we're just going to make a G ranges where the start and end are the summit. So it's essentially the same place. And then we'll resize this by 100 and fix it in the center. So we'll essentially take that G ranges where we're essentially just focused on one spot and extend it 50 base pairs on each side. So now we'll have a G ranges, which is centered 100 base pairs for each peak, centered around the summits, which should be the best input for motif enrichment, and that G ranges is shown here. Once we've recentered our peak and we have the G ranges in place that we like and that we want to use for motif enrichment, we'll use the getSeq function, which will return a DNA string set object containing, it should be as long as your G ranges containing the sequence, the DNA sequence underneath those G ranges peaks. So we just use the getSeq function with the BS genome object for the genome we're interested in, and then the G ranges that we want to extract the sequences under or from. And we'll just give these sequences sensible names, and we'll just give them the names, as you can see here, of a combination of chromosome and start and end, which will be a unique identifier for each row. Then the write X string set function will allow us to take this DNA string set object and just write it as a FASTA file. So this will just be a FASTA file where each row is the sequence from each peak. And so this is the input that we can use for Memechip. And Memechip can be used, we often use it locally. You can download the software and use it in the terminal if you're comfortable with that. But there's also a web portal where you can just input your sequence here, and it'll, I don't know, maybe 5 or 10 minutes later, you'll get results, and it will look something like this, where you'll have the motifs that were identified. So these are de novo motifs, so they were identified independent of a motif, and then you then compare it to known motifs and get a p-value that's based on how many of your peaks actually had this motif in it compared to what you'd expect by chance. I do know that Max is a common cofactor of MIC, and so that's good that came up, and it's very highly enriched, and some other ones come up. So this is sort of a common workflow for to identify other types of motifs that are enriched within your peaks. One of the outputs from MemeChip, this is a FEMO output file, which is like a GFF3 file, or it's a GFF3 file, but which you should be able to visualize in IGV, but for some reason this GFS file naming conventions don't allow a lot of them to be reported. So we can use our track layer, import this GFF file with the import function, give them more sensible names, and then export this back out again with cluster profiler with the export.gff3 function. And now you can see we have a lot of these motifs are showing up. And so this is showing us just where the motifs that this isn't necessarily the MIC motif or the Max motif. This is the actual motifs that MemeChip found for us. So we can see where they are. We can zoom into our favorite gene and see if our gene that we're interested in actually has one near it, or whether a certain peak we're interested in has one of these motifs. And so it allows you to explore the data a little bit. There's a biostrings functionality where we can actually scan our sequences and look for the occurrences of certain motifs, of known motifs within our G ranges. So we're not necessarily, in this case, finding a de novo motif, but we're determining whether a known motif occurs within our peaks. And there's various databases that do this. One common one is called Jasper, and it's tended to and updated every few years. The most recent update was 2020. And there's also a nice R bioconductor package to load the database in with just the Jasper 2020 package. So we're loading it in. We can look at it. It's just a database. This is the path to your local computer that has the database once you install the package. We can use the TFBS tools package, and specifically the get matrix by name function, where we can use this function, specify Jasper, and then give the name of a motif we're interested in or multiple motifs. And it will pull out what's called a PFM. Sometimes you'll see PFM or PWM. PFM is position frequency matrix and PWM is position weight matrix, but they're just different ways of showing the likelihood or frequency of each nucleotide at each position along a motif. So it's that link that we clicked on for MemeChip. You know that logo plot where the bigger the letter is, the more likely it is to be in that position motif. That's usually generated from a PFM. So with the PFM or PWM, you can use another package called Motif Matcher, which will scan the peaks that you give it with the G ranges and return the positions of the motifs within those ranges. Or you can also change this out parameter here, and it'll tell you you can get a counts table, which will just be the counts, so the number of motifs that are in each peak. So there's a few different options there. Here we're just returning the positions of the motifs that fall within our peak. So you just give it the PFM, which is the location, or which is the frequency matrix of your TF of interest. You give it your G ranges, the out parameter, and the genome, the FASTA, the BS genome object. And the output is a G ranges list that again has all of the positions of all the motifs that overlap your peaks. And we can export this with export.bed from our track layer and get a bed file where we can then go in IGV and look at the actual positions of the motifs that are overlapping our peaks of interest, or our peaks in general. So as usual, there's exercises that you can do on your own time and ask us about anytime, either now or on GitHub or via email. But yeah, so I think, so that's it for now. We're back here at 3 o'clock for one more session on taking, so now we've just been working on one peak set. We'll move on to taking the peaks from all 4 of our replicates and trying to sort of make meaningful conclusions about those based on consensus peaks and non-overlapping peaks, and then finish with differential ChIPseq. So as usual, I'll be around now and until 3. So just speak up or send me a message on the chat, and I'm happy to answer any questions. And if there's no other questions now, then I'll see you in a little bit. But again, just please feel free to ask questions if you got them. I'm glad you all stuck around. I appreciate it, and hopefully you've learned a lot. So this is our last section. Again, we're going to look at looking at our replicates as a whole and trying to identify high confidence peaks, also identifying unique and common peaks at certain conditions, and then using that common set of peaks to see how we can do some differential ChIPseq. So again, it's a similar cell line or similar samples. This is mixed ChIPseq from the MEL and CH12 mouse leukemia cell lines. Links for each cell line to download the relevant files are there. If you downloaded the sample materials, these peaks files will be in the peak subdirectory of the data file, or data directory, sorry. And so as we've been over throughout the day, obviously the goal of ChIPseq is to look at how your factor or epigenetic mark is localized not only in one particular place, but genome-wide. And by doing that, you can hopefully understand something about the biological role of your protein of interest. And so as ChIPseq has developed, there's been a wide-scale mapping of transcription factor binding sites or epigenetic states. And all this is, most of this is cataloged on ENCODE's website. Sorry. And also the advent of multiplexing, so the ability to run or sequence multiple samples with the use of different indexes in the same sequencing lane has allowed us to try and sequence more samples at once, potentially get replicated ChIPseq experiments. And then further things, I'm sure some of you have done some things like cut and run that have tried to even reduce the numbers of reads that are required for each ChIPseq experiment to try and increase our ability to do more replicates. Because as much as we want it to be, ChIPseq isn't the most quantitative method. It is to some extent, but it really, really benefits from replicates, not necessarily to identify the peaks, but especially to try and do the differential peak call or differential signal that we're going to look at today. And it just increases confidence. And so, like I said, you know, peak calling really is a binary. There either is a peak or it isn't, and that can heavily depend on what peak caller you use and what you're really looking for, whether it's broad or more punctate peaks. And, you know, this is kind of an example. We're here in the middle. You know, this is a peak that's kind of binary. It's here in this cell line and not in this one. But then over here, you have some peaks that, maybe look like it's reduced in this heart sample compared to this top one, but you obviously can't just look at that by eye and be like, oh, this is reduced. This is something I should look at for this particular gene. So we're going to look at how we can try and assess whether things like that are different when it's not so clear, like there's a peak or there's not a peak. So peak will be called in every sample here, but you're not sure whether it's significantly different or not. So the first thing we're going to do is try and define, use some of our bioconductor tool set to define high confidence or reproducible peak set, or a set of peaks for our MIC ChIPseq experiments in these two cell lines, and also identify unique or common between the cell lines too. So using the same peaks, now we're finally going to delve into all 4 peak sets at once instead of looking at one replicate at a time. So we want to read in all these files, and first we will just get a list of the file paths, and we can do that with the dir function pointing to a particular directory, which in this case is the peaks directory in our data directory. You can select, you can indicate a certain pattern that you want, and it'll match, or it'll pull out the paths for all the files that match that pattern. And this, we've done grepping, but essentially the star means any number, dot means any character, and then you want to end with, or to have, at least to have peaks in it. And then full names true is going to give us the full relative path. And so when we run this function, what you see is that you get the entire path for all 4 of our peak sets, or peak samples. And these are these outputs from max2. So once we have these paths, we're going to make an empty list and then loop through and read each file from the paths that we just made. We're going to exclude the comments at the top. So if you remember in these output files, there's a number of lines that have some helpful documentation, but you can exclude them because they all have the comment character in front of them. So at the end, we'll have this list, and each element of the list will be a data frame that's read in from these max2 generated peak files. We then want to convert these to G ranges, and we've done this before a couple times today. In this case, we're just going to do it in a loop. So looping through the list of data frames, we're then going to make a list of G ranges. So for each element of the list, we're just going to convert it to G ranges with this G ranges function, where we just build the required components, which is seq names and the start and end within this iranges function. And just to reiterate, we did cover, you can use the import function and you'll get a G ranges as well. This is an R-track layer function. And this narrow peak file is just another output file that exists after you run max2. So in the end, we have a list of G ranges. Here we're accessing the first element, which is just going to be whatever the first set of peaks that we read in. We'll want to give them sensible names. So we'll use some base commands to do that. So we have this is our vector of paths, which are the paths to the peak files. We can use this base name function, which will just give us the file name, and it will exclude everything upstream of that in the path. And then we can use the gsub function, which basically looks for the pattern in your first argument in any element of the third argument, and then replaces it with this. So we're essentially just looking for this peaks.xls and replacing it with nothing. And the result is just what we want, which is nice, sensible names for each of our replicates. And then we can eventually use these names. But first, we're going to convert our list of G ranges to a G ranges list. That sounds very similar, but it's a subtle difference. So we're basically just taking our list, which is a regular list that just happens to contain G ranges, and we'll apply this G ranges list function. And naturally, what this does is make a G ranges list, and it may not seem super different, but as we'll see in a little bit, as a G ranges list, you can usually apply certain functions that are used on single G ranges to these G ranges lists, and they wouldn't work on a regular list. So it just allows you to use these nice bioconductor functions to interact with it in a list format. And we'll see some of the benefits really soon. Then we're gonna take those names that we made in the previous slide and make these the names of the lists, because otherwise we won't know, unless we keep track in our head which element or which number, we won't know which one corresponds to which replicate. And if you look at the class of this list, you can see it's a G ranges list, whereas had you looked at this, it just would have been a regular list. So as I said, G ranges list can often both act like a regular list and a G ranges list. Here we're applying the length function to our G ranges list, and we get a nice output that tells us the length of each G ranges, which the length of each G ranges is just the number of elements or rows that it has. And so this is essentially the number of peaks we have within each of these replicates. In addition to having these being able to use common list functions, we can use specialized G ranges functions on these lists, these G ranges lists. And this is nice because there's no otherwise you'd have to do an lapply, which would convert it to a regular list and then go back to a G ranges list if you want to use these sort of common G ranges functions. But this just keeps it all in one type of nice G ranges object. And so here, for example, we're applying the resize function, which if you remember just takes your G ranges. If you apply to an individual G ranges, it'll just resize it based on the parameters you give it. Here it's going to go through this entire list and resize each of them individually to be 10 base pairs centered around starting at the center of the peak. And then you can also, you can use this width function and you'll get a nice output because it's a G ranges list, which just shows, clearly our function has worked. Our widths are now 10, and you can easily see it for all 4 replicates that because it's a G ranges list, you can just use these simple functions and get a lot done really quickly in just a few lines of code. And so now that we have a list, we can also, if we ever want to use each one individually, we can easily just extract it with a normal, you can use the double brackets, or you can also use this $ accessor as well. And here we extract mel1 and mel2 peaks, the replicates individually, and you can look at their length, do anything you want to it. We can also use, we can easily find unique, or we'll see in a second, common peaks. For the unique peaks, for example, we used this earlier, but we can use this over operator, which goes through each peak in the first thing on the G range on the left and tells you whether each of those ranges overlaps with anything in the G ranges on the right. So it gives you a logical vector. Here we're going to take the opposite because we want the unique. We don't want the ones that are over. We want the ones that are not over. And so here we're getting the peaks unique to mel1. Here we're getting the peaks unique to mel2. You can see how many. You can export these very easily with our track layer, this export.bed function. And then you have something that you can look at in IGV. And here's, obviously we're doing what we want. Here's a mel1 specific peak up here. These are peaks that are common, that are present in both, so it's not going to be present in our mel1 or mel2 unique bed file. We can extract peaks that are common to replicate 1 or replicate 2, and that's just by simply removing that exclamation point. So you're actually filtering and keeping the ones that overlap. You can export those again as a bed file, and it's always good to confirm an IGV. to prevent bugs. But you can see here we're excluding this one that's unique, and we're pointing to and isolating the peaks that are common to both. You'll notice too that the numbers of common peaks is not identical. And the reason for that is sometimes, you'll have multiple peaks in one that overlaps with one big peak in another, and so you'll get different numbers of common peaks. So for example, in this case, here we're contributing 3 peaks to the common peak set, and here we're contributing 2. And so you're depending if one sample has broad peaks and one sample has a number of sharper peaks that overlap that, you're going to get different numbers of common peaks. And so oftentimes you want one common set of peaks across a set of samples so that you can then study them, for example, for differential peak calling. And so defining a common consensus peak set is something that you'll often want to do, and we're going to go through how to do that here. And so to do that, what we're gonna define our consensus set will be a non-redundant set of peaks. And so basically we want any point of the genome for which a peak in any sample occurs, but we don't want any region to be in our GRanges object twice. And so it's pretty easy to do that. The first step will be use this unlist function. And if you use unlist on a GRanges list, it basically just flattens it. So it takes all the elements in your list and essentially just stitches them together almost as like an R-bind. And then once you have that, so this set's going to have, it's not doing anything about combining peaks. If you'll have lots of peaks that overlap within the genome. So you can use this reduce function on your unlisted GRanges. And what this will do is it basically just lines up all your peaks, and if you can imagine, in a linear fashion across the genome, and just combines everything. So if you have 2 peaks that overlap on the end, it's going to combine that into one long peak, and in the middle where they overlap, it's just going to collapse that into one range. And if two peaks were to perfectly overlap, it would just convert that into one peak. And so here, after the unlisting and reducing, we now have a non-redundant distinct set of peaks representing peaks in any sample, either in MEL cells or in CH12 cells. And this is our non-redundant peak set here at the bottom, the black bars, and you can see this is what we've picked out. Here's some CH12 specific peaks, they're included. Here's some CH12 and MEL very close together, but they're all picked up, and then these are MEL specific peaks. And so everything is included in this peak set. So now that we have this newly defined non-redundant peak set, we can go through and pull out sort of interesting combinations or pretty much anything we want in terms of overlaps with our specific sample. So here, for example, we're identifying peaks that are in both MEL replicates. So we're subsetting the whole non-redundant peak set with those that are over MEL1 and operator and over MEL2. And so here we're getting a common set of peaks for MEL, and it's one number. It's not like before where you have to choose whether you do MEL1 over MEL2 or MEL2 over MEL1. This is a nice consistent way to get one common set or one set of common peaks. And you see we've done that here where we pull out the blue as our MEL1 common peaks, and it's looking at just the MEL1 specific peaks. We can obviously do any combination of these. We can just also look at those that are present in only one replicate. So over MEL1, not over MEL2, not over MEL1 and over MEL2, and you get your specific ones. So this is, similar to what we had done before, but this is working off one common set of peaks, which gives you It's just a different result, but it's potentially a little more consistent across types of samples. And as expected, here we have our MEL1 only, which are those that are only here and not here, whereas these peaks don't pop up. For more complex overlaps, or to be able to catch them in sort of a more programmatic way, it might make sense to define a logical matrix which describes which samples are present in our non-redundant peak set, or for each non-redundant peak, which samples that peak is present in. And so to do that, we're going to create an empty list, and then we're going to cycle through each of our G ranges in our G ranges list and create this. This will be a vector, a logical vector. So we're not subsetting anything. We're just creating a vector that will say whether the peaks in our non-redundant peak set, whether each peak overlaps with any peak in that particular element of the list, which in this case is one of our replicates, either MEL or CH12. And so if you look at one element of this overlap, again, it's just a logical vector, which is the result of this over operator here. We can then make a nice matrix of these vectors using the do call, which will basically, which is a nice function that can basically cycle over a list and apply the function, the first argument to that list. And so it's going to go through overlap and C-bind all of the elements of overlap together. We'll set the column names of our new matrix to be the names of our G ranges. And so now we have a matrix where every column is a vector of overlap. So the rows are the peaks, columns are samples, and each element will tell you whether that particular sample has a peak, has an overlapping peak in this peak from the non-redundant peak set. And so then we can, as we've done before, we can use the mcalls accessor to then stitch or attach this matrix as metadata to our non-redundant peak set. So this non-redundant peak set is the G ranges, remember. And so we can put this overlap matrix, insert that into the metadata, and the result is this nice G ranges that has all the information of our particular range, and then whether that range is present in any of the 4 samples. And so we can easily then identify peaks that are, present with certain combinations, and we'll in a second sort of use this to really tease apart or identify peaks that we're interested in for our differential peak calling. This type of matrix here is amenable to a Venn diagram. The lemma package is also commonly used for RNA-seq and microarray data for a lot of reasons, but one useful function it contains is a Venn diagram function, which allows us to plot these overlaps from a logical matrix. So we can just do Venn diagram on the mcalls, which is just that logical matrix, and we can see this. 4 samples can get a little overwhelming, but you can see some things right off the bat that this particular replicate has a lot of unique peaks. I think it just has a lot more reads, and as you'd expect, you have a lot of overlapping reads for the replicates. And you even have a bunch that are common amongst all 4. So you can really get some quick information about what's going on just by making a Venn diagram, and it's really easy here in R. Lemma also has this Venn counts function, Venn counts, which just takes the same mcalls, which will just be a data frame, and will return the counts. So these are just the specific counts that went into that Venn diagram. So for example, here's those unique peaks to this CH12 number 2 replicate, which had a lot more reads that we could see in the Venn diagram. Okay, so before we move on to differential ChIPseq analysis, we want to define one set of peaks and what we'll call our high confidence peaks. And to do that, we want peaks that are present in both replicates of each type of sample. So one way we can do that is, since the logical matrix that we appended to, that we put on as in the metadata of the G ranges is essentially equivalent to a 1 or a 0, because 1 equals true and 0 is false for any logical matrix. We can use the row sums function directly on this matrix and use the sum of the rows, either a 1 or a 0, to extract the peaks that have at least 2 of the CH12 replicates, meaning that it has a peak in both CH12 replicates, or within this non-redundant peak, there is an overlapping peak in both replicates. And so we'll subset our non-redundant peak set. This is the G ranges, remember, by, there's a lot going on here, but I guess we can take it one step at a time. So this is subsetting this mcalls, so subsetting essentially this matrix here by the 2 columns for CH12. So we're essentially just taking these 2 columns here, turning that into a data frame, because row sums only acts on a data frame, and calling row sums on just this data frame here. And so if row sums equals 2, meaning if there's a true in both spots in that particular row, then that peak will be kept and put into this G ranges object here. And then we can export that as a bed file. And you'll see, as expected, we've pulled out this peak here, which has peaks in both replicates, whereas here where we have peaks in replicate 2 but not replicate 1, we leave those out. We're not considering those high confidence peaks. We can also do things like identify the high confidence peaks that are specific to CH12. So here we're using the same idea to get those that are present in both MEL2 replicates, but row sums of the MEL2 section of this metadata equals 0. So there's no peaks overlapping, and this will give us the CH12 specific peaks. You can export that, look at it, and it's going to be, again, this same one because there's no peaks over here in MEL. So you can see, you can quickly really identify, and your scientific question might be this: you want peaks that are present in this one but not in this one. And this is, once you get to this point where you have this matrix appended to your ranges, there's a lot you can really do. And the fact that you can easily just export this as a bed file and quickly check it out on IGV and cycle through and visualize it is really nice. And you can really get through some data really quickly to get an idea of if something interesting is going on. And so now that we have our high confidence, non-redundant set, well, I guess we don't have the high confidence yet, but we have a method to get high confidence peaks. But we have our non-redundant peak set. We know how to interact with it and really manipulate it. We're going to try and do some differential analysis. And so this comes into play for lots of reasons. As we said, peak calling is binary. It's a peak or no peak, and that can be highly dependent on the peak caller. And you might get situations like this where, for example, here and here, you might call a peak. You probably will call a peak in both samples, but it does clearly look like in this Pol2 resting on the bottom that there might be an increase in signal in this resting state compared to the cycling state. Whereas if you just looked at peak calling, this wouldn't come out as anything interesting because you'd probably have peaks in both. And clearly, there's something different between these two peaks and this peak here, where they look exactly the same. And maybe if you'd had corresponding RNA-seq, you would notice that these 2 genes are highly expressed in the resting state, whereas this is unchanged. And so you want to tie your ChIPseq changes or your RNA-seq changes to ChIPseq. You wouldn't see that in peak calling here, but you might see it if you do some sort of more specific differential analysis. And so essentially what we're going to do is it's going to be very similar in the end to your RNA-seq, where in the end you just want a counts matrix that is, instead of samples by genes, it's going to be samples by peaks. And so we need to get a counts matrix of samples by peaks. But to do that, we need a common set of peaks over which to count fragments from each sample, IP'd fragments. We need to get that one set, and what we're going to develop is use our non-redundant peak set to get a high confidence set of peaks that we can use across. And so what we're going to, what you can decide whatever you want depending on your sort of threshold of sensitivity, but for us, we're going to be pretty stringent, and we're going to take peaks that occurred in both replicates of either sample. So we want high confidence for CH12 or high confidence for MEL2. And so we're going to use the same row sums approach where we want the row sums of the CH12 samples to be 2, or the row sums of the MEL1 replicates to also be 2. Not also, but or. So this will give us, again, high confidence peaks in either cell line. And that's what we're pulling out here, where here's a high confident CH12 and here's the high confident MEL, but these ones in the middle that are only present in one replicate for each are left out. So now that we have our regions over which to count, we will use our BAM files, which, if you remember, are just our aligned reads. So we'll quantify the number of reads that overlap each of these particular regions, and then we essentially will have a counts table that we can use for differential analysis. So we've used this function before, this BAM file list, but it's part of the RSAMtools package. So we'll load in RSAMtools. We will get a, we'll insert in the path to each of our BAM files. And then we'll use this BAM file list function, which takes the path to our BAMs and a yield size, and we'll read in this number of reads at a time. So it allows us to cycle through this BAM file. And this will go until the BAM file, until it uses all of the reads in the BAM file. And so to count the number of fragments overlapping our peaks within our BAM file, we're going to use the summarize overlaps function. And ChIPseq is generally strandless. So we're going to set one of the arguments is ignore strand to be true, and we're going to set that to be true. So that just means it doesn't matter if a read's on the positive or negative, as long as it is within that range in our G ranges, then it's going to count. And so with the summarize overlap function, we give it our high confidence peaks. We give it this BAM file list object, which will tell it how to cycle through and how many reads to look at a time for each BAM file. We'll ignore strand, and after a bit of time, the result will be this range summarize experiment object, which we've seen before, is essentially, it's a common bioconductor object that will have our G ranges in it, and in the assay slot. So this is kind of similar to the differential expression that you guys did yesterday, will be the counts table in that assay slot. And so it's basically just a big object that allows us to access different parts of the data depending on what we want to do. And we can, because this, oftentimes when you do a function that takes a long time, you can save the output as an RData file, and then you can just load that in next time instead of having to, like, if you want to come back and do some sort of different type of analysis you did the first time, you can come back and just load in this RData file and start from there. So that's, I do that a lot for not just things like summarize overlaps, but any type of R object that took a while to produce, especially once you're dealing with things like Fastq and BAM files. So to assess the changes in signal across the cell lines, again, this is going to be pretty similar to differential expression of RNA-seq. We're going to use the DE-seq2 package. And there's multiple ways to do this, but we like DE-seq2. It fits very well in with the bioconductor framework. But it contains a workflow for assessing local changes in fragment abundance, which is similar to counts in an RNA-seq experiment. And it takes care of normalizing, variance estimation, and removing outliers. It does some very nice estimation of dispersions and variance depending on whether your region has a high number of counts or low number of counts, and it's just very well suited to NGS data. So as part of the DE-seq2 workflow, you need to have a data frame. You might have remembered it yesterday as your call data. So it's column data, so it's data that's describing things about your samples or your conditions of interest. So the row names of this data frame need to match your sample names, which will be the column names of your counts table, which in this case is our BAM file. And then your columns will be different descriptors of these particular BAM files. So here our main condition is cell line. Let's say you had a treatment, you could have another one for treatment or time frame or whatever you want. But here again, we just have one thing we're looking at, which is the difference between cell lines. And then we can use the DE-seq dataset from matrix function, which is just one of these constructors to get it into this format that DE-seq needs to do the analysis. And there's lots of DE-seq constructor functions, but since we just have essentially a counts matrix in this summarized experiment object, we can just use that and build the DE-seq dataset from there. So our count data is going to be the counts table of our summarized experiment object, which you can access with this assay function. Our call data is going to be the data frame we just made in the last slide. And our design has to be one of the columns in this metadata data frame, which in our case, we only have one, so it's going to be cell line. And then there's an optional argument for row ranges, which will then include the row ranges, which is just your G ranges of your high confidence peaks within the object, so that you can later tie or link certain ranges to whatever the statistics that come out of your differential analysis. Yeah, so again, we're testing based on cell line and using the counts data frame or the counts matrix from our summarized experiment overlapped object. So then we take this DE-seq object, yesterday I think it was called the a DDS, and we run the DE-seq function. And this does all the size factors, dispersions, size factors meaning like how many, it's usually tied to sequencing depth. Dispersion estimates, it applies your model. And here we're assigning it to the same name. This is, you can assign it to a different name if you want, but in this case, it actually works out pretty well because the DE-seq function doesn't overwrite anything in the object. It basically just adds to it. And so if you were to look at it before and after, you'd see afterwards there's some new things, like some of these statistics that weren't present before are present now. But the most important thing you can do with this object after you've run the DE-seq function is extract information about our differential regions using the results function. And so you can use the results function, the first argument being a DE-seq2 object that you've run the DE-seq function on. The second argument will be the contrast, which is going to be a vector of length 3, where the first element is the metadata column of interest, and then the two groups that you want to compare, because sometimes you may have multiple groups more than 2. And so you can select the ones that you want to compare to or compare against. The output format we're going to set as GRanges. And so you run this function. It's pretty quick. It's very quick. And you get a data frame like object that we're going to order by p-value. Well, actually, in this case, it's a GRanges because we specified it to be GRanges. But one of the columns is p-value. So we'll sort it by p-value. So our most significantly changing regions will be at the top. If you look at the class, it's the GRanges. If we actually look at it, this will look a lot like the data frames you saw yesterday for RNA-seq, but here, since we specified GRange, it's the GRanges, and we have our range, and for each range, we have the statistical comparison between whatever we chose. In this case, it was obviously MEL versus CH12, and we have our log fold change and our p-values. And this is what we can use to pick out differential regions. And this is what we're doing here, where we're filtering this output, this results GRanges, first getting rid of NA, because some of the, in this results table, you'll often get NA for especially really low expressing genes in the case of RNA-seq, or really low peaks that don't have many reads in them, where you couldn't really do a appropriate statistical test. So they're usually left in there, but they have NA and their p-value. So we like to get rid of these because NAs can break lots of further downstream testing, and we're not interested in those ranges anyway. And so we can get up and MEL, which are those that those peaks. We're going to subset our GRanges based on those that significantly changed and those that had a log 2-fold change greater than 0. And then down in MEL is the same thing, but the log 2-fold change being less than 0. And we can export these as bed files. And as we usually do, we go and look at them. And this particular region, again, we're just seeing these peaks where there was actually just a binary yes or no, but you could, which is the CH12 specific here and the up in MEL here. And so, if you went through the genome here, you'd actually see places where you have peaks in both, but it's up or down in MEL. And that's potentially more interesting, and you pull out more regions than you would if you were just looking at overlaps or those that are in common. One cool package is the track tables package, which you can take a GRanges, put it in this make bed table function, give it an output HTML and the directory to which you want to put it, and it will produce this nice table here where if you click on this IGV link and if IGV is open, it'll bring you to this particular genomic region. You can also search things, and you can scroll back and forth. And so ideally you have IGV open with your big wigs loaded, and you can click on this link and go actually see what this position looks like and see whether you're actually convinced that it's a region that is significantly changing, or whether it's near genes that you're interested in. You could take this GRanges now and annotate it with ChIPseq as we saw before, and then you can then link those genes to significantly changing regions, or you can take the significant ones and go and do our gene set enrichment that we did in the third lecture. So this allows you to kind of get to a point where you can sort of more with a fine-tooth comb go through certain gene sets and understand what's going on, and potentially make better hypotheses for your future experiments. Okay, so that's the end. So again, there's more exercises. I highly recommend you do them. You really won't learn this stuff until you sort of chew on your own data and dig in a little bit, or dig in a lot. But again, ask us questions once you do exercises. We're really excited to answer them and work with you guys. And yeah, thank you all for sticking around towards the end of the day. I appreciate it. Hopefully you enjoyed it. And yeah, I'll be here. Again, I'll probably be around for the next half an hour or so. on this call if you guys want to ask questions or ask them now. But yeah, thanks.